kind of put Chris's slide up first, because he's the one with slide, and he's gonna spend, he promised me 2 minutes.
I got some slides too.
Oh, you do? Okay, Mike, good, all right. So, so we're just gonna go quick 2-minute slide, just to level set kind of maybe some of the backgrounds of the companies, which some of you may not be familiar with. We'll go with Anpac Bio first, and then we'll go to Diacard second, 2 minutes each. And then we'll let each of the other participants speak for 2 minutes. So that'll take 10 minutes out of the 45, and hopefully leave ourselves 35 minutes to get into the debate. So with that, you want to put up Chris's?
Okay, good.
Chris, here you go.
Okay, good morning. I'm Chris of Anpac Bio.
I don't know if you have any, I don't know if it's one slide or two, that'll
Okay. We are an international life science company with operations in both US and China, but we have 200 patents filed in 20 countries and regions with 100 patents issued. The company was founded in 2010, so it's not really a new company. And the project started in 2008, so it's been 10 years. Just give you some summary. Our business is focused on cancer detection with utilities in all 5 major categories. And current focus is in cancer screening. But we also are working with many hospitals on prognosis and reoccurrence. And so far, we have served 140,000 individuals. And last year, we had a paid test of 50,000 individuals. This year, we will have over 100,000 paid tests, and we forecast 500,000 individuals for next year. And what is unique about technology, in contrast to traditional liquid biopsy, is our signal is biophysical-based properties, which gets into the inside of multiple levels. And where the company championed the idea of a multi-level, multi-parameter idea, which you may have seen the Johns Hopkins paper earlier this year in Science, which talked about a combination of mutations with the protein biomarkers. But in 2015, we already proposed and showed results from 4 types of different cancers. The second major idea we championed is multi-cancer testing. If you go back to look at the literature and announcement, we were the very first company actually proposed the multi-cancer screening, and with the first technology, first product developed. And so far, we are already screening individuals with over 26 types of cancer, and with a large database. And more importantly, we are saving lives on a weekly basis. So Anpac is not a laboratory phase. We are already commercialized in Asia, and we are screening 80 days, hundreds to thousands of individuals a day, and with paid tests. And so far, we have screened out close to 400 individuals with cancer. Most of them are pre-cancer or stage 1 cancer, and with very good results in terms of early stage cancers being screened out in stage 1. For example, for non-small cell lung cancer, our sensitivity is 85%, specificity is 90%. We believe we set a world record at this point for non-small cell lung cancer in terms of specificity and sensitivity. I think I have kicked off. I'll go to the next one.
Chris, would you mind if we kind of, I think the audience got a great flavor for it, but given the number of speakers, I think we might want to just jump to Michael and Diacard, if you don't mind. Is that okay?
Okay, can I just say one word about this one? Okay. So basically, this shows our technology, actually the signal starting tracking at healthy. So the biophysical properties we're testing already in existence with the healthy individuals. So that's quite different than traditional liquid biopsy, which starts at when the solid tumor is formed. So our signal starts from healthy state, progresses through disease state, pre-cancer state, and to stage 1. Thank you very much.
Great, thank you.
I'm sorry, taking a little bit longer.
Great. So can we go to Michael Powell and Diacard?
Yeah, I'll go so fast.
Okay, that's all right. Okay.
So like we were founded in 2011, and
You can't see the world on here, but we have a lab, the biggest sequencing lab in the world in Nanjing. We have 60 NovaSeqs in that lab. We're part of the Million Genome Project in China, and we have another lab. Those are the labs we have in China. We have 200 people in China and 30 people here in the US, where I'm based in San Francisco. And all the inventive stuff's done in San Francisco, and we do all service business in China. Next slide. And we have all clinical certifications. This is what we do. We basically block all the wild type and amplifier news. And we got a lot of awards. There's all these articles that came out. Next slide. We can enrich. This is what we do. We enrich and we increase the reliability of NGS, and the cost goes right down. So we don't do deep sequencing. We can get all the we take all of the low frequency mutations, make them germline before you put them on the machine. So we're trying to democratize the process too. Next slide. And we have a LiquidBiopsy test for colon cancer that can find adenomas from blood. We look at 22 mutations, 11 genes. This is a real-time PCR assay. It's a kit that works on all these platforms. It's approved in Italy, and it's going to get FDA approval here soon. Next slide. And this is another test we have. It's the only test of its kind in the world for doing companion diagnostic testing for radiation therapy. Because how do you know when to stop irradiating a patient? And this has been matched with the MRI imaging, and it's funded by the NCI. We have a contract to make this FDA-approved test. Thanks.
Great, thank you very much. Thank you for the updates. That was very quick and fast. So I do have a number of questions, but what I'd like for each of the participants up here, because each of your businesses is very different. And maybe we can spend a couple of minutes, try to keep it tight and discuss, kind of what your technology is, kind of what market you're serving, because I know each one of you are going after different kind of applications and target markets. So.
Sure. So as Epic Science do, we bring a bit of a contrary view, I guess, to a genomic conference. So I guess our thought process is that while I think there's an increasing acknowledgement cancer is a disease of the DNA, the functional unit of the disease to our mind is actually the cancer cell and the clone. And so while genomic drivers are obviously very important, it's I think becoming more and more evident that you have epigenetics, you have the proteomic layer, but ultimately all of those things come together in the clones that the disease spits up, and then also how those clones evolve as we give sequential round of treatment. So when we look at liquid biopsy, really what we're doing is looking at all nucleated cells in a patient's sample, and then using a combination of staining, computer vision, machine learning, to not only find the rare cells, in other words, all the circulating tumor cells that may be present as a proxy for the overall metastatic burden of a patient, and which are really the most pernicious actors. But the majority of cells we actually analyze are the leukocytes. So as we think of this new world of immuno-oncology, we can get a profound parallel view of not only how a patient's cancer has evolved clonally, but how his immune system responding or not responding to particular therapy we throw at them. So we've really created, we think, an incredibly powerful platform for biomarker discovery. And then we've mated that now to actually clinical cohorts that we've gone out and selected very diligently with a number of academic partners. So contrary to trying to essentially assemble panels that we can apply across all indications, what we've gone to is specific therapeutic decision points. And we've asked physicians, where do we have the most clinical leverage, in the sense that if you get this therapy wrong, then that's likely to be the last therapy a patient gets. Versus if we can actually get it right, we can significantly extend life in a meaningful way. And if we did a test that could help get that treatment decision right, then we have a large enough addressable market to create meaningful products. And so we've been able to collect patient samples before, during, and after treatment, biobank them with a proprietary process, and then once those patients hit their clinical endpoints, unblind and say how well do the biomarker signatures our platform identified correlate to overall survival under different treatments, right? And I think we got to go to overall survival and life extension, and not go to purely prognostic signals. So we've done that. The first test that we've commercialized now on an exclusive basis in partnership with Genomic Health is our ARV7 test in prostate cancer. First predictive test in metastatic disease of this sort, 100% positive predictive value. And so we're very excited about that. But then we're now also building a pipeline of tests that are looking at all of the key drug classes in pharma's development pipelines now, such as PARP inhibitors, IO drugs. And lastly, we're also beginning to decipher when as a patient's clonal diversity reaches such thresholds that no targeted therapy may actually work, right? So understand that clonal evolution. So it's really, I think, a powerful mix of a novel discovery platform that's looking at the functional units of the disease and then correlating to heart clinical endpoints.
Great, thank you. Great.
I think Freenome might be the youngest company on the block here. So we're based in South San Francisco. Raised our seed round in 2016 and our Series A last year. And our goal is to really make a difference in the mortality of cancer by detecting and intervening in cancer at its most treatable stages. So to that end, our primary development program is actually in cancer screening, specifically for colorectal cancer. And the thing that really sets us apart from the way the rest of the genomics field is looking at cancer detection is that most people are focused on the notion that cancer is a disease of the DNA, that there are going to be DNA alterations released by cancer cells into the circulation that you can find. The challenge is that literature has shown that particularly in stage 1, about half of patients have so little cancer DNA floating around in their blood that you'd have to draw about half this Fiji water container in order to even get a single molecule out in your sample. So the approach that we're taking is instead of trying to delve signal out of the 0.1 or 0.01% of tumor-derived DNA, try to find signals from the host's response or the immune response to the presence of a tumor in the other 99.9% of the cell-free DNA, as well as looking at other signals in the blood, things like proteins and RNAs. Using this multi-analyte technology, integrating it with AI and machine learning to basically use the body's response to cancer as a natural amplifier for the intrinsic signal challenges of detecting this. Now, the nice thing about this, you can imagine that such a technology could have many other applications beyond colorectal cancer. So we have programs in line of sight towards developing screening tests in other indications, as well as partnerships that we have announced, exploring opportunities to use this technology in other areas of biomarker discovery, not just cancer screening.
Great, thank you. John?
Thank you. So at last evening's pharmaceutical session, it was mentioned that the ultimate one day would be to develop a drug that would be targeted at each patient's mutations. And that's not one day. We're doing that today at Personalis. We do that with our customers who are pharmaceutical companies. Personalis is the genomics front end on the clinical trials for almost every clinical trial in that space today. So literally a patient's tumor will be analyzed. We'll sequence it, DNA and RNA, full exome level, identify the neoantigens. We use mass spec data that we have accumulated with a machine learning approach to be able to rank the neoantigens in terms of their antigenicity and to recommend which ones would be appropriate targets, whether it's for a personal cancer vaccine or whether it's for a neoantigen targeted CAR T therapy. But these are therapies where the FDA has already approved clinical trials. We've been handling samples for patients in those clinical trials for the last 2 years. And this is a worldwide effort that's being sponsored both by large pharmaceutical companies. There have been announced programs by Merck and Amgen and Roche, but there's also at least a dozen small biotech companies, and most of those folks are our customers. Most of them are in the U.S., but an increasing number are in Europe. So that's essentially what Personalis does. It has become noticed that the impact of these neoantigens is not only important when you have a drug that is explicitly targeting those, but those neoantigens are impacting the outcome of the patient, even if you're using a drug that's like a checkpoint inhibitor. And so what matters is not just how many somatic mutations you have or what your tumor mutational burden is, but which neoantigens actually will be antigenic and what the clinical effect of that will be. I think the biggest thing that genomics can do to improve outcomes for cancer patients is not just to do more screening, but to help develop drugs that will actually target the mutations that most people have. And we think that neoantigen targeted therapeutics will be the future for that. Just as a rough financial perspective, Personalis has been in business since 2011. We have about 125 employees. During the life of the company, we've received about $120 million in customer orders. Over 70 million of that's been in the last year, so the company is accelerating relatively rapidly. We have a fairly large sequencing lab, and we are, as a secondary business, we are the whole genome sequencing engine for the VA's Million Veterans Program. So far, we've been contracted to sequence 45,000 whole human genomes for that project. So that's what Personalis does.
Great, well, thank you for all that. So I have the challenge of trying to find questions that just cut across 5 different platforms, but I set the panel off. So No, I think some of them will definitely be specific, but maybe one question which I think a few people alluded to on the dais here, certainly ARV7 is already on the market and Personalis as well, but in terms of the COGS, of kind of your test and how you thought about developing a test that's going to achieve what you wanted to, at the same time make it affordable. So can you just speak to kind of pricing and cost and whether or not a common theme that we've tried to explore in different panels is as the price of sequencing goes down to the extent you are sequencing, how does that impact, how does that change what you're doing? So maybe we can just go right down the wall so we can start from there.
Well, our technology is platform agnostic. I mentioned a couple of products there. We don't care what instrument works on all instruments. We work on the chemistry, the upfront chemistry. So our coloscape assay, we don't take a whole Fiji bottle of blood. We actually, we eat 4 mils of plasma. That's what we do for our clinical studies. We can find low frequency mutations because of our chemistry. Because we enrich all of the oncogenic mutations. That's how we find them. We have a technology that allows us to do that. Other people don't have that, so they always make that statement, we're never going to find anything. We actually have done clinical trials and clinical studies with cancer patients, early stage, late stage, and we find mutations with people with early adenomas. We can do that. That's why we're pursuing this very aggressively. The cost is standard PCR. It works on all PCR machines. So you've already got the machine in your lab. Just buy the kit. The kit is half the price of the Roche and Kaiser kit right now. It's clinically approved in Europe. We sell it in Europe. We have a lung cancer test too. For the OptiSeq panel, we're developing nano panels. As I mentioned, we don't need to do deep sequencing. You can put 50 samples on a MySeq with our technology. As you know, if you do deep sequencing, you can only probably put 1 or 2 samples on a MySeq. And our turnaround time from plasma to result using OptiSeq is 48 hours. We don't need a huge amount of bioinformatics analysis because every low frequency mutation in your patient sample, by the time you put it on the MySeq, it's germline. Because we block all of the wild type. We have a chemistry that does that. So our cost is very low because we're not, you have to buy any new hardware. The turnaround time is much faster, so you're not investing in so much time, and it's much more reliable. The reliability goes up. So you don't have to do any orthogonal testing, like to see whether it's a real mutation or not.
Okay.
Yeah, so let me take the question in 2 pieces, right? One is what happens if sequencing costs go down 10x, and does that impact us? Well, we try to make a distinction between access to technology versus clinical utility. So we're focused on clinical utility, and so while we think it may be helpful in some circumstances if more people are sequenced, ultimately we're trying to give docs actionable results that are very binary. So for example, in the ARV7 instance, doc gets a plus or minus result. They want to see cell images, they don't know how to interpret them. But if it's a plus, it's very simple. Your patient is no longer will be responsive to anti-androgen therapy, must be switched to chemotherapy. You get a minus, you can cross-switch the targeted therapies and still receive clinical benefit. Now, the beauty of our platform, we think, is it's imaging-based. So as a result, broadly speaking, we believe we can get COGS advantages of 80 to 90% reduction in COGS versus comparable sequencing-based tests. And even in some instances where in an R&D phase, we're looking at genomic phenomenon in the cells we find, what we've actually been able to do is then use that computer vision technology to say, hey, did all the cells that had a particular genomic phenomenon we're tracking happen to morphologically exhibit in a specific way? And then convert that into a morphologic signature that now can be run at high volume at exceptionally low cost. So that's enabled us, in essence, to have a COGS profile where we can have really powerful commercial partnerships and even post those revenue splits, come in and have medical device and tools margins, the 70+% range, as an example.
Okay. Mm-hmm.
I totally agree with Marley's take on clinical utility, right? The goal here really is clinical impact. The way I see sequencing prices, it's as though we're talking about cars and gas prices, right? Does a falling gas price or electricity price, help both Honda and Ford? Yeah, absolutely. Same way, falling sequencing costs are going to help anybody who's running a sequencer. Data that we have disclosed at AACR this year, we've talked about our low coverage whole genome sequencing assay. I think we're potentially less bound by sequencing costs than some other folks, but really in the end, the price is going to be set by the marketplace, and I think there are benchmarks that you can look at in the market to see where people have landed on what the reimbursable price is for screening, and we're going to aim to launch something that's commercially viable.
Okay.
So we think that sequencing cost is important for what we're doing for the future, but we're aiming at drugs that may not be approved by the FDA for a few more years yet. So we think there's time for those costs to continue to decrease. Our tests tend to be quite expensive because we're doing a lot of sequencing, but we think it's important to get high quality data and to build the applications around these new drugs that we see coming to market in the next few years here. So just to give you an idea, sequencing on a single patient, when we work with cell-free DNA, we might sequence several 100 billion bases of sequence. When we work from tissue samples, it might be somewhat less than that. But they're pretty big assays, and so it is helpful for us that the cost is coming down on the scale, going from what we used to look to as the $1,000 genome to what you put out there as a $100 genome. I'd say Illumina's already made meaningful progress in heading from one to the other. They're not quite there yet, but I think we're heading there, and I think it will open up the ability to look at all the neoantigens in a tumor, not just a few hundred driver genes, which is kind of where we are today.
Chris?
Because Anpac Bio is vertically integrated, intrinsic cost is very low because we design, patent, and assemble our own machine and getting certifications. And so our cost, we believe, is one of the lowest in the industry. And for the US market, we target to have an ASP of $260 per test for individual test for 6 most common types of cancer. So that's a test kit to screen for 6 types of cancer for only $160, which has significant market impact, especially in terms of this low price point allows some other payers, especially insurance companies, which were already in serious discussions and with one of the largest insurance companies in the country. We also have surveyed a large number of employers in this country, and some of them already indicated once we get LDT approved, which will be in the next 6 months, they will pay directly to Anpac for their employees get screened. Of course, we're also going to file 510K in Q1 of next year, getting FDA approval.
Okay. So I just want to come back a little bit just so I can clearly make sure that we're getting at the real differentiating technology, and maybe the patent estate of technology each of your companies is kind of the point. So maybe we could start with Mike. It sounds like it's this ability to look at the mutant versus the wild type in terms of the captures. Maybe just speak to a little bit exactly what, is it the chemistry, am I right in that regard? Discuss your patents around that and kind of, what does that really enable you to do?
Yeah, well, as you know, right, when you take a blood sample, you're looking for 2 copies of the mutant, oncogenic mutant, and 10,000 copies of wild type. So all the other guys do deep sequencing. You have to sift through the haystack to find. What you do is you drop a PCR, you spread it all out, and you say, now you can see the mutant. What we do is we develop a method to burn the haystack and make a stack of needles. That's the simplest analogy. We just replicate the needles and we block the haystack. And we have 80 patents filed on the use of, it's called XNA. And some of you may know about PNA and LNA. And well, XNA is LNA and PNA on steroids, okay? Because there's limitations with LNA because you're going to use small amplicons to block it.
What is it, LNA and PNA?
Locked nucleic acid. PNA is peptide nucleic acid. So I was in Peter Nissen's lab in 1992 when he invented PNA. And these are blockers. And another company uses oligos with a phosphathyroid technology called CAST PCR. You may be familiar with that from Thermo Fisher. But they use an oligonucleotide blocker. But that's not very efficient. So I invented XNA. So I'm a chemist. I used to make protease inhibitors for Roche in my whole life. So I know how to change backbones. So that's what XNA is, a modified backbone. It has no phosphate, no sugar, but it still has nucleic acid bases on it. And I can make you any sequence of this XNA to any target. I can make mutant-specific XNA, or I can make wild-type specific XNA. And I can also make chimeric XNA. I can link XNA to other stuff. For example, you've seen all the big news about CRISPR being a problem, big problem, all these off-target effects. Well, if we take XNA and link it to guide RNA, all those problems go away. So our patent just published on that, so you can find that. We also use XNA and CRISPR. It's the fastest way to screen for CRISPR genetics because you're looking for very tiny amounts of mutation. It's the best way to find mutations because you enrich the mutation and you can detect it any way you want. You can even do a dipstick assay if you want, because it's all there. You make billions of copies of mutants, and there's no wild type amplification at all. That's been the major problem. The other problem with blockers is it's non-soluble. PNA is not very soluble, and it only works for certain sequences. GC rich, you can't do it. Purines won't work. XNA is like DNA. You can make it to any sequence, and it's universally available, and you can block things, or you can use it as a probe. You can even use XNA for in situ detection of mutations, which is a big thing. You're always talking about mapping things in tissue. We can actually find mutations in tissue using XNA, because we link XNA with branched DNA. We have another technology, the diacorder. Our other technology is signal amplification based on branched DNA. You may remember this from Chiron. Some of you may be old enough to remember the company Chiron. Got bought by Bayer, then Siemens took it over. And we're the only company that has what's left of branched DNA. Affymetrix has branched DNA 2.0. We have branched DNA 3.0, which means there's no background, because all the branched nucleic acids are made on a natural basis. So we can combine XNA with ddNA. Now we can find mutations in situ. And people are interested in that, because when they do CAR-T engineering, they want to see if the tissue has what they're looking for. So that's another application. So we have patents on that too.
Okay, great. Ron?
So coming from what was historically, we'll call it a circulating tumor cell space, right? The challenges initially were that people make assumptions about what these cells in circulation are supposed to look like. They were all supposed to have epithelial proteins on their surface, or they're all supposed to be a specific size. So a lot of what I would call Gen 1 companies use engineering approaches to try to solve the problem. They'll use magnetic enrichment or some kind of size-based filtration or gating. And in fact, many approaches still do. What Epic kind of did was something quite orthogonal. They said, well, we don't know what we don't know yet. And we know we have a very heterogeneous disease, and that's been one of the reasons it's been so tough to manage. So let's actually take a shotgun approach. Let's not select on any basis, but look at all nucleated cells in a sample and use that combination of staining, computer vision, and proprietary algorithm to really find the rare event. And we could do that because the majority of cells we look at, as I mentioned before, are leukocytes. So we know what normal leukocytes look like, and they're the vast majority of cells. So anything that deviates from that average in terms of morphology and or staining, and really the combination thereof, you can flag as a rare event. And then you go and have the luxury of being able to say, is that a rare event a biological sideshow, or is it clinically meaningful in terms of understanding response to particular drugs? So from a first principle basis, it's just a completely unbiased approach, and lo and behold, these cells are actually not as rare as we thought, and they're far more different species than we thought. And so now we've been going systematically in understanding how they're correlating to different patterns of drugs. So as a result of that, now we have over 65 biopharma partnerships, and one of the key advantages of our platform is that we can take that 10 ml blood tube, it arrives at our door up to 72 hours post-phlebotomy, and we can lay out all those nucleated cells across a series of 10 to 12 proprietary blast slides. So these kind of have a surface coating, we liken to molecular Velcro. All the cells sit in an unbelievably even monolayer, and we can then biobank them, store them in a -80 freezer, essentially indefinitely. So we've created an FFPE equivalent from the blood for all intent and purpose. And then we want to test either for our biopharma work or for routine clinical testing. We can take any subset of those slides, stain them, image them, and then understand really what are the rare events. And now really the next phase of this is actually going into IO, where we can not just characterize the rare cells, but sub-characterize the different leukocyte populations present. Because we believe actually the biomarker may not be something singular like tumor mutational burden or PD-L1, but maybe a combination of cancer clones and T cells or other types of immune cells that together create a higher order pattern that now we can start to link to clinical response. And if you don't have a platform like ours, which can actually see all of this in parallel, I think it's going to be incredibly hard to find maybe some of the more nuanced patterns that may be there.
I think the unique thing that we bring, the thing that I think will really change the ability to solve this problem, is a tight integration across different disciplines, and really that tight integration with laboratory technology and molecular biology, but also the computer science and machine learning downstream. So to get more specific, we're doing really interesting things, I think, with multi-analyte assays in the wet lab, where we can look at multiple things from a single tube of blood. We can look not only at cell-free DNA, but we can look at epigenetic marks, We can look at proteins, we can look at circulating RNAs as well. The challenge with this is that, what this gives you is a huge amount of data. So, you may have more information there, but now you have a real big data challenge of dealing with this downstream. This is why you have to have tight collaboration between the laboratory scientists who are developing these assays and the computational scientists downstream who are actually developing the AI and the machine learning to be able to pull patterns out of this data to actually recognize what's happening in the body. As Murali was pointing out, we're probably not going to be in a situation where the biomarker is exactly one gene or exactly one mutation. There's going to be a combination of things that, as I mentioned earlier, are collectively informing you about the body's response to the presence of a tumor. So only by actually being able to discover these patterns, but being able to discover these patterns leveraging the unique laboratory technologies, are we going to be able to make progress on this? Now, I think the really interesting thing that machine learning technology gives you beyond the discovery phase is the possibility for real improvements even post-market. One of the best known features of machine learning systems is that they tend to get better as you feed them more data. So obviously you're going to feed them data during development, you're going to feed them data during validation. But I think a real opportunity that the biomarker field hasn't really tapped to date is the ability to integrate large-scale real-world evidence, post-commercial launch, accumulate that data, potentially stage out new revisions to a test that are able to just further increase your ability to deliver performance. So Dan, to answer your question, it's not really one thing. It really is a combination of being able to deeply understand what's going on at a molecular level in the sample, but really then being able to put that together with the analysis upstream.
Okay. John?
Yes, so I think historically, looking at cancer, we would have focused on what we call driver mutations, and in fact, we thought those were the good mutations to pay attention to, and other things might have been considered sort of dismissively as passenger mutations. We now know that those passenger mutations are the vast majority of the mutations, and although they don't necessarily cause the cancer, they may be the things which actually flag the immune system to attack the tumor. And so part of Personalis' technological differentiation is that we built everything from the ground up to look at all of the genes. So whereas other groups may look at 300 genes or 500 genes, Personalis, in everything we do, looks at all 20,000 genes. We do that on tissue biopsies, and we do that on liquid biopsies. So that's probably the first differentiation. The second thing is then, once you've identified the genetic variants that create those neoantigens, you need to identify which ones could be antigenic, and so you'd be interested in knowing which one of those peptides could be bound to the patient's HLA type. That's a complicated problem today. We use machine learning algorithms for that, but the key thing for machine learning is not the neural networks, because we have those and other people have those. The real issue is what's the data that you can feed into that. And so we've developed over the last several years a proprietary set of cell lines where we've done monoallelic transfection. We put a single HLA allele into each cell, and then we've developed mass spec data on the peptides that are actually bound by those HLA types. So that then lets us train our neural networks to be able to recognize of the perhaps large number of somatic mutations, what we would have considered passenger mutations in the past, which of those might actually lead to peptides that can be bound by that patient's HLA and potentially provoke the immune system. And then that can lead to the therapies for that patient that are based on those neoantigens. So those are some of the technical differences. I'd say on the regulatory side, part of what's different is because we're in drug clinical trials, everything we do has to be approved by the FDA along with the trial of the drug. So Personalis work has been approved for multiple FDA-approved clinical trials. We've been through that over and over and worked with the FDA. We've also been approved in New York State and have a Anpac laboratory as well. So I think there's the regulatory side. And then I'd say, just from a financial standpoint, our strategy has been different because we've been working with on therapeutics, which we think are actually one of the biggest challenges here. We work mostly with pharmaceutical companies. With pharmaceutical companies up front, we don't have a reimbursement problem. We've been paid for 100% of everything we've ever done. So the company is heading towards profitability and is actually already cash flow positive. So I think it's a different situation. Eventually, if these drugs are approved, the drug companies will have a reimbursement issue, but typically drug reimbursement is quite a different environment from diagnostic reimbursement. And so we're participating in the upside of the pharmaceutical space, but without the binary risk that you typically might have if you were a biotech with a single drug where your clinical trials either going to succeed or fail.
Chris, what's your technological secret sauce?
Good question. traditionally, cancer diagnostics or screening focuses on one level, either molecular level, genomics, we're talking about here, or protein level, or cellular level, for example, CTC. And 10 years ago, myself and Professor Herbert Yu at that time at Yale University, and we joined, we have taken a much deeper fundamental approach. We're thinking, finding a set of fundamental properties, biophysical properties, which will tie all those levels. I'm talking about molecular level, I'm talking about cellular level, and a protein level. What if this set of properties reveal not only their individual contributions to the signal, also their interreactions, which we never thought about. Cell talks to each other, right? Cells talk to each other, cells talk to DNA. So we have explored so little so far, but we have taken a totally different approach. So using an integrated system in which you are able to probe something we have never probed before. And by doing so, we find this set of properties occur in a normal individual. And as we track it, go from a normal individual to disease, and from disease to precancer, precancer to stage 1, we find that statistically, the signal from this set of properties is significant, statistically meaningful. And so by doing so, we believe we have shed some new light. Because along the way, if you think about it, we always wonder, even though we know some reason why T cell at some point lost the surveillance capability, lost ability to kill cancer cells, we never truly understand it, why it's happening. But by probing this along the way, we find something very fundamental. And our experimental data actually has shown. And for example, when we looked at the signal from control group to disease group to cancer group, the difference is significant. In fact, if the curve I showed to you earlier, if you plot the slope, actually the change point is really from disease to precancer, from precancer to stage 1. We believe that's the way to really do early stage cancer screening. Having said that, we also worked with a large number of hospitals and did targeted drug treatment, how we do the follow-up testing, how we follow different drug treatment. For example, in one hospital, we collected data on AstraZeneca's one of the targeted drugs for non-small cell lung cancer, which also shows it's very useful. So what we have developed is a platform on which we are tracking dynamic progression from a healthy individual to disease state, from disease state to precancer, which included polyps in colon and nodule in the lung. If I did a predal analysis in what we have actually screened out, prospectively screened out patients with precancer conditions, we find that the top 2, one is polyps in colon, the second one is nodule in the lung. And so this technology platform is fairly broad, can be used for different utilities.